Hi everyone, welcome to my channel Pure Biology, and I am Taposhi, and this is the continuing part of mitochondria. And before this part I have already discussed about the mitochondrial structure and related another topic of mitochondria obviously. And this is the third part of the mitochondria. Here I try to discuss about the TOM and TIM. Actually, TOM and TIM, the introduction of these two TOM and TIM, what that already I have described in the previous video. So you just check out that one, then continue with this. So this TOM and TIM, both the translocases that have to cooperate tightly to promote protein translocation. Okay, so in the protein translocation, both this TOM and TIM have a major responsibilities. Okay, and this TOM complex, it consists of 7 different subunits. Okay, and the receptors TOM20 and TOM22 and TOM70, these help to recognize precursor proteins and transfer them to the central component of the channel forming TOM40. So what is saying here, that the channel forming protein, that is TOM40, and not this is the actually the mitochondrial channel forming protein. Okay, TOM40. And what is happening here, the receptors, actually this TOM generate TOM containing 7 subunits. Among these subunits, TOM20, 22 and 70, these can recognize the precursor proteins, okay, which become mature to form the proteins. Okay, precursor proteins, that's why these are called the proteins, not in the mature form. So these recognize this protein and transfer this protein to the central channel. Okay, and where already present another protein, that is the TOM40. Okay, so actually these TOM proteins or the subunits of the TOM, so these, what is the function? These actually recognize the proteins, precursor proteins, and transfer them. Okay, these are the actual function of this TOM and subunits. And there are another 3 small subunits, TOM5, 6 and 7. These are involved in the assembly and the dynamic of the TOM complex. So TOM complex formed by these 3 subunits, 5, 6 and 7. So actual function of these subunits or this complex is to identify or recognize the protein and transfer them to the central channel, clear, of mitochondria obviously. And here the 2 TIM complexes, this 23 and 22, the role is that transfer the different proteins. Okay, so here 50, TIM 50, that acceptor function in intermembrane space, or you can say that TIM 50 is present in the intermembrane space, and here these are responsible for transporting the precursor proteins. And TM23, that present in the channel forming, present in the channel area, and these help to form the channel forming proteins. Okay, and here the TM50, actually these are responsible where the intermembrane space where the proteins are there, and these proteins are totally transported by this TIM50, and 23 that are responsible for the channel protein, okay, or channel forming proteins. So these are the TOM and TIM translocases. Okay, actually this contain many subunits and its role is just identify the proteins and then transfer to the channel area. Next is the mitochondrial targeting sequences. There are some mitochondrial targeting sequences obviously present. Here, the mitochondrial proteins that translocate from cytosol to mitochondria that contain mitochondrial targeting signal sequences. So, only with the help of these TOM subunits or TIM subunits that proteins are not transported. So here the some signal sequences obviously responsible in that function. Okay. So there are two signal sequences. One signal sequences is known as classical N-terminal cleavable matrix targeting sequences or pre-sequences, and another is non-cleavable internal targeting sequences. Clear? So two kinds of sequences are there. One is the classical N-terminal cleavable matrix targeting sequences and another is the non-cleavable internal targeting sequences. Two kinds of sequences, signal sequences obviously, or you can say the mitochondrial targeting signal sequences are generally present. And these pre-sequences are rich in positively charged basic amino acids, not any negatively charged amino acids. Pre-sequences means which pre-sequences? This classical N terminal cleavable matrix targeting sequences, these are known as the pre-sequences. These are actually containing the positively charged basic amino acids like that arginine and lysine, and some hydroxyl group containing amino acids also present there. These are known as the serine, example is serine or threonine. But negatively charged acidic residues absent. So pre-sequences have the positively basic amino acids and hydroxyl group containing amino acids. Clear? And typical pre-sequences, generally the length of the typical pre-sequences is 10 to 30 amino acid residues. It containing about 10 to 30 amino acid residues. And these are the precursor proteins. Okay. So which precursor proteins? That is related to this precursor proteins. Okay. So these precursor proteins. So these are the actual sequences of the precursor proteins that are either cleaved or non-cleaved way transported by TOM and TIM. So it's actual relation of this and this. Now here the where that precursor proteins are where present, these are the precursor proteins and generally present in the mitochondrial matrix, inner membrane and intermembrane space. In three areas the precursor proteins are there. Clear? And this is cleaved once it reaches the matrix. After, when it is cleaved into the short fragments, then it enter into the matrix. Okay? And it is removed by the mitochondrial processing peptidase. Mitochondrial processing peptidase. When it enters into the matrix region after fragmentation, then these are processed by MPP or mitochondrial processing peptidase, and that resides in matrix. That resides in matrix. A large number of protein contain that NCITS. What is NCITS? That means non-cleavable internal targeting sequences. These include all outer membrane proteins and inner membrane space and inner membrane proteins. So under these non-cleavable sequences, there are many proteins can remain. One is outer membrane proteins, maybe inner membrane proteins, or maybe intermembrane space protein, but maximum is the outer membrane proteins. Okay, and these are obviously non-cleavable. So what is the gist of that? That first TOM and TIM, these contains many subunits, and its responsibility is to recognize the precursor proteins and then transport them to the channel area. Okay, this is the channel. Where is the channel? Channel forming. Okay. Okay, so this is the TOM and TOM and TIM, and both containing many subunits. And what are the sequences? The precursor protein sequences. These precursor protein sequences generate two type of. One is the cleavable, another is the non-cleavable. The cleavable that containing the basic amino acids and the hydroxyl group containing amino acids. Okay, and obviously the positively charged amino acids, not negatively charged amino acids. First, these are fragmented into the smaller part, then it enters into the matrix region. Okay, and then it is removed by the mitochondrial processing peptidase. Clear? And what happened to the non-cleavable? The non-cleavable, not here described properly, just saying that non-cleavable proteins, these generally containing the outer membranous proteins, and here the main difference between the cleavable and non-cleavable, that means the cleavable proteins is fragmented, then after that the processing is possible, and here in non-cleavable proteins there no fragmentation is seen, just these are processed. Okay, so these are the actual topic related to the mitochondrial targeting sequences and TOM and TIM. And next, another topic, that is targeting of mitochondrial proteins into mitochondria, or the targeting to the inner membrane, how it is possible, I will try to describe in my next video. Okay, so continue with me and my channel.